Hi, welcome back. This is still Unit 3.2, Structure and Function of DNA. However, we are at our last section, Section 5. Woo! So Part 5 is on mutations, cancer, and asexual reproduction. But this is our last section of this chapter, so let's go ahead and get going, shall we? All right. So mutation, what are they? How do they work? Well, we're only going to kind of cover just the basics of them right now. But remember, DNA is your genetic code. It's your blueprint. And it is what will determine all of your characteristics. And that's because they determine the characteristics of your cells. Our DNA is filled with genes. Genes are a section of the DNA that are used to then create our proteins. Now, there can be a mistake made during DNA replication or during division that results in a change to the actual DNA-based sequences. And when there's a change like that, it's called a mutation. A mutation is a change to the actual DNA-based sequences. Let's see. As you can see with this snake, it has a mutation where the pigments on some of its scales aren't showing through. That's the result of a mutation. So what are they? As we said, mutations are changes in the DNA code, and typically this can either result from an error occurring during DNA replication, or it can be damaged by some kind of environmental factor. Now, when we talk about environmental factors in terms of biology and genetics, an environmental factor is anything outside of your genetic code, so anything outside of your DNA. Whether it's your diet, your lifestyle, or anything like that, things you're exposed to, things that you eat, all of that is part of environmental factors. Let's talk a little bit about inheritance. So, can mutations be inherited? Well, yes, some can, and well, some can't. It all depends on where the mutation occurs. If the mutation occurs in a gamete, in a sex cell, then it will be passed on to the next generation. In that case, it is inherited. So if there is a mutation in the sperm or the eggs of a woman, then after fertilization occurs, that mutation is present in the zygote's DNA, which will then be present in the baby's DNA, and it will be present in every single cell of that baby. However, if it's a mutation that occurs in a somatic cell, which is any other kind of cell besides sex cells, if that's the case, then it won't necessarily be passed on. Now, I give you some examples of both of these. In the young lady on the left-hand side, she has a mutation where her leg is actually backwards. I think starting around the knee portion, but her leg formed backwards. That is actually part of her DNA, and she can pass that on to her kids as well. As opposed to something like down here on the bottom right, this is melanoma, is skin cancer. Now, the reason why that can't necessarily be passed on is because, let's say I go out and I spend a lot of time out in the sun. You know, it's summertime right now, why not? Well, too much sun exposure, the UV light can actually cause damage to your DNA. So what we're saying can happen is maybe this cell right here, the DNA in it gets damaged by the UV rays from the sun. Now it can be damaged, it can grow and become skin cancer. Well, that bit of skin cancer right there is not going to affect my eggs. So I'm not going to pass it on. It occurred because the sun affected this one cell right here. It damaged this one cell right here. This cell divided, and now I got skin cancer right here. Unless it breaks off, goes into my circulatory system, and infects my ovaries, it's not going to be something to worry about in terms of passing it on. And even then, it won't necessarily affect my eggs because my eggs are already made. What it will do, though, is it could affect my ability to have kids if it damages my ovaries. So that's an example of a somatic mutation, and meaning it's one that occurs in my body cells due to damage from an outside source, not going to be passed on. I did want to present this example. It's called the tree man disease. And what it is called, the actual name for it, epidermal dysplasia eruciformis. Now what happens is these thick plaques start to build up on the skin, and they just keep building up and building up and building up. And over time, since they build up on top of the existing plaques, they start to thicken out and get this almost like tree trunk appearance. Now his hands are not that big, but they're hidden beneath layers and layers and layers of this plaque that's building up on his skin. You see how originally it started off like this, then they keep building up and building up until it's like that. Now they can be removed, but it is very painful from what I've read, and also they keep growing back. So even though, yes, you can get these removed, it's gonna grow back again and again and again. Here's the x-ray. You can see the bones underneath, like there's a finger bone, and then you see the plaques on top of the skin. And they will cover the entire body, as evident by, you can see it's up the arms, on the feet and extremities, see a little bit on his neck, and all over his face. Now that is an inherited disease. It is in his DNA, and should he choose to have kids, it will be passed on. That gene will be passed on. Now, if it's a recessive gene, it doesn't mean that his kids will necessarily have it. They'll just have the allele for it. If it is a dominant gene, though, then his kids, yes, they will have it. All right, there are three main types of mutations that we are going to mention. The first is called a point mutation. And this is where a single base is changed, so just one single base. For instance, let's say in this 3-base sequence, this is found in your DNA, AAT, but there's a change and it changes to ACT. That's an example of a point mutation. Let's look a little bit more into this. So here's the original DNA, ATG, which leads to the mRNA, UAC, which leads to tyrosine. So that's the amino acid that would be normally placed into the protein and have a functional normal protein. Now, if there's a mutation in it, depending on where it's located, it may or may not affect the protein. If the last one is changed, remember it's ATG. If the last base is changed to ATA, gives us the codon UAU, it still results in tyrosine. So even though there was a mutation, we call this a silent mutation because it didn't affect the protein, so it didn't create a change to the expression of the trait. ATC instead of ATG, that leads to UAG, which leads to a stop protein. Now, a stop codon. If this occurs anywhere but at the end of the mRNA, then that means it's going to stop transcription and translation early, which means your protein is going to be short. And it might just be short 1 or 2 amino acids. It could be short 1,000 amino acids, in which case it's going to have a serious effect on its ability to be a functional protein. Let's see. What if the first one changes? Instead of ATG, it's GTG, leading to CAC, and that codes for a different amino acid. That codes for histidine. Now, the function of a protein is based on the exact amino acids in an exact order, because the placement of the amino acids in the chain will determine how it folds up. And how it folds up determines its function. Well, having a different amino acid in one location can cause it to not fold up in the correct way, which means you'd end up with a faulty protein. It's not doing the job it's supposed to. And again, that can have pretty serious complications and effects. sickle cell disease or the sickle cell disorder is actually the result of a point mutation. One base is changed, changing one single amino acid in the chain, and that affects the hemoglobin protein. Next, we have frameshift mutation. These are where there's changes in the DNA due to either adding extra bases or deleting bases. And what that's going to do is going to cause all the bases after that to shift over, causing the codons to shift, so it could mean that they are different. Here's an example. So here we give you 3 codons, AAT, CCA, GAG. Now what if in between these first 2 A's, an extra base is added, maybe a cytosine? So it gets added there, it pushes all of the other bases down, which is going to affect the order of the codon. So now we have ACA, TCC, AGA, and then this lonely little G out there. So here's some examples. In the normal DNA sequence, here it is, and it creates the, going through transcription for the mRNA and then translation. It ends up with the protein or the amino acid sequence of tyrosine, histidine, proline, lysine, and valine. Now, if there's an insertion, meaning you insert a base, we're gonna put it right here between these 2 C's, and we're gonna add a guanine. That's gonna cause everything to shift over one spot. So we end up, the first 2 are the same, but now we have CGC instead of CCT. The T got pushed to the next codon. And that's going to change the amino acids. We end up with an arginine and a stop codon. So this is actually going to be shorter than what it should be. Even if it wasn't, it'd probably be a different amino acid. And that's also a different amino acid. Plus, a single base doesn't code for any amino acid. All right. Well, what if there's a deletion? So we lose a base. This adenine gets taken out, but that's going to cause everything to shift in the other direction. So TAT, instead of being CAT, the adenine was deleted, so that shifts the T and the C from the next codon over. So we have CTC, CTA, AGG, and then these 2 leftover bases. Now that's going to change everything. Look, remember it's histidine, proline, lysine, valine. Now we have leucine, leucine, arginine, and then nothing there because you need 3 codons, 3 bases to make 1 codon. 1 codon makes or codes for an amino acid. If it's 1 base or if it's just 2 bases, it cannot code for an amino acid. And last is chromosomal mutations. So these are changes to the entire chromosome. So it's not going to affect just one gene. It can actually affect multiple genes by leading to changes in the order of the genes. So we give you some examples here. Here we duplicated the section, so that made the chromosome twice as long, or this section twice as long, so it elongates the chromosome. Here, we flip the parts around, so we invert them. Here we are showing a deletion to an entire section of the chromosome. Here we're showing crossing over occurring between 2 non-homologous, well, sorry, that's not crossing over. This is when a part of this chromosome is broken off and inserted into a completely separate chromosome. So part of this one moves to that one. It's called a translocation. This is when we have crossing over occurring between 2 non-homologous chromosomes. So the genes on this chromosome get exchanged for the genes on that chromosome. Now in all these cases, it's putting genes in the wrong spot. Our cells are taught by the DNA how to read the chromosomes and how to look for particular genes for particular traits. So if we're moving them around, then we can't necessarily find the correct genes, and that can have a serious effect on whether or not the gene gets expressed and if it gets expressed correctly. It was just kind of showing you another version of that chart. A, B, C, D, E, F. In the deletion, you're going to lose something. So whether it's 1, 2, 3, 10 genes, you're going to lose a section of the chromosome, and that can include multiple genes. So A, C, D, E, F. A, B, C, D, E, F. With a duplication, a section of the chromosome, a copy is made of it and placed right next to the original. Now again, this section could be 1 gene, it could be 5 genes, it could be 10 genes. The point is that we copied the entire section and left the copy in. An inversion is when it kind of flips part of itself around. So remember, we had DE on one side and BC on the other. Well, now we have ED, so the order of them flip-flop, and they're on the wrong side of the centromere. Same here, CB. So B came first originally, now C is first, and they're now on the opposite side of the centromere. Here to read this backwards and look correctly, B, C, D, E. What we did was we took this section here and flipped it around. Or you can have different kinds of translocations. This is showing the translocation that's a crossing over between two non-homologous chromosomes. We have A, B, C, D, E, F on one and G, H, I, J, K, L on the other. But what happens is with this particular type of translocation, crossing over occurs and they exchange information that is not homologous. So we end up with A, B, C, J, K, L, G, H, I, D, E, F. So because we're changing and affecting the order of the chromosomes, that can actually have a big effect on how the genes are expressed and even if they are expressed. So effects. Well, a lot of times we like to mention effects in terms of being bad, but that's not always the case. Mutations actually can be good. I mean, that's the whole basis of natural selection. When there is a mutation in the DNA that actually allows an organism to better survive. For instance, in this bottom picture on the left, we have the two birds. One of them has a mutation that makes the beak longer and thinner. Well, now it's better able to survive because it has an easier time getting food. So that means he can live longer and have more baby birds that have the same mutation, the long, thin beak, as opposed to this bird, which can't get as much food. It has a harder time getting food, so it's going to die sooner. He's not going to have as much babies born. And over time, the population will shift so that all the individuals have this new trait. Skin color is actually the basis of, not the basis, it is a result of a mutation to better enable humans to survive. If you were to look at a map of the world, which I actually do have, I just don't have it in this PowerPoint, but it looks at the origin of different skin tones. And what you find is that the darker the skin tone, you tend to see that trait originate closer to the equator. And the farther you get away from the equator, the lighter the skin tone is. Well, it's actually because of, again, survival. Individuals whose ancestors hail from the equator regions, they were exposed to a lot more sunlight. Around the equator, there's a lot more direct sunlight of the entire year. So what the skin does, the skin creates this pigment called melanin. And that's what gives the darker coloration. The more melanin your skin produces, the darker your skin is. And what it does is it actually blocks a lot of the harmful UV rays. Now, yes, we do need exposure to some sunlight and some UV rays because they do help us build things like vitamin D and work with calcium. But we don't want too much UV radiation and sunlight because it can also lead to skin cancer. So the excess melanin, which leads to darker pigments, help because that melanin will actually block more of the harmful UV rays. But as tribes moved away from the equator, then let's say the tribes that ended up in Northern Europe, there was less direct sunlight throughout the year because of the tilt of the earth. The sunlight more so comes in at an angle, and you don't have consistent sunlight the entire year round. So what happened is the skin started producing less melanin. Still some, because we still wanted some protection, but not as much. Because now that those tribes, and yes, my ancestors as well, hailed from areas where there was less consistent sunlight, they needed to absorb more of the UV rays in order to make vitamin C and help with calcium absorption and all that. So we need to absorb more sunlight, hence less pigmentation. So all that is a result of pigmentation and the amount of melanin being produced based on survival of your ancestors. Now, yes, some mutations can be bad and they can lead to different diseases or disorders. Down here at the bottom right, I show an example called porphyria, or it's a variation of porphyria. And porphyria itself is a metabolism error or mutation. There's a mutation that causes an error and prevents someone with it from being able to metabolize certain minerals, so they can't actually get those minerals from their food. As a result, you tend to see sores and lesions forming. They can actually end up having organ failure because they're not getting the required vitamins and minerals to keep their organs going. But that's a mutation that would hurt the survival of that person. If we weren't talking about humans, if we were talking about something out in nature, if you notice a lot of animals and plants, we don't see as many diseases or disorders that affect them. And that's simply because since it hurts their chances at survival, oftentimes they would die before mating and having offspring, so that gene for that trait wouldn't get passed on. But in humans, because we have medical advancements and we can care for ourselves and for others, we do tend to see a lot more diseases and disorders because we're able to treat them or at least help make things better for the individual afflicted with it. And some mutations can be neutral, meaning they just simply provide variation. They don't hurt your chances of survival, but they don't help you to survive either, like having blue eyes. Blue eyes are the result of a mutation that occurred in the human genome about, give or take, 10,000 years ago. Now, does blue eyes hurt your chance of survival? No. I know this because I got blue eyes. They don't help me to survive any better. It's just simply a variation in the phenotype, in this case, of the eye color. So mutations can be good, they can be bad, or they can be neutral. Oh yeah, I gotta move my camera back. All right, so now let's talk about cancer. So remember, we said that in the cell cycle, after division, cells go to interphase, and they're able to check for errors and then just do their normal function, their normal job. There's actual mechanisms, both chemical mechanisms within the cell as well as mechanisms outside the cell that will regulate how a cell should grow, to what size it should grow, as well as when it should divide and how often it should divide. Well, what we found is that certain mutations, or mutations in certain parts of the DNA, I should say, actually affect the cell's ability to regulate its own division, meaning it can grow faster than it should be, which will then lead to division at a much quicker rate. Now, this is actually what cancer is. It's unregulated cell growth. but because it's unregulated, it can have a lot of severe effects. So cancer is a disease that results from unregulated cell growth. Now, sometimes they can be caused due to things that we are exposed to, such as certain chemicals like teratogens. Chemicals in cigarettes have been linked to lung cancer. So constantly exposing your lungs to this particular chemical found in cigarettes, over time, it damages the cells and the DNA in the cells of your lungs, leading to lung cancer. Asbestos can, over time, too much exposure to it can also damage DNA and lead to something called mesothelioma. So these chemicals, we call them carcinogens. Carcinogens are specific teratogens. Teratogens are chemicals that can damage growth and development, and carcinogens are ones that specifically lead to cancer. They can also be environmental conditions that can lead to cancer. So for instance, we mentioned being exposed to too much UV light from the sun can lead to skin cancer. UV rays can actually damage the DNA in your skin, leading to this unregulated cell growth. Air pollution has also been linked to certain kinds of lung cancer as well, because of the toxins within the air pollution. Now, in some cancers, there are what we call genetic predispositions, meaning that there are some genes that we have found linked to cancers, and we found that in certain families are more likely to develop these mutations because of DNA replication in errors occurring in it. But what we found is that certain of these mutations are more likely to occur in certain families. And again, it's just based on the genetics. It doesn't mean you will get that cancer. It just means that you are more likely to. So for instance, when a woman goes to a doctor, one of the questions they always ask is, does anyone in your family have breast cancer? If so, who? And what they're looking for is a trend. Does any of your immediate family members, like your mother or sisters, have it? Do any secondary relatives, like an aunt or cousin, or even grandparents? Or is it more further removed? The more further they are removed from you, the less likely that you would have the same genes that led to them getting breast cancer. If it's your mom or your sister, you're more likely to also develop breast cancer. It doesn't mean that you will. It just means you have a higher probability of having the genes that will cause that unregulated cell growth within the breast tissue. Now let's move on to asexual reproduction. So you guys know humans are sexual reproductions. That means that are sexually reproducing organisms, which means that there is an exchange of genetic material between two parents. So the offspring from sexual reproduction are the result of the combining of DNA from a mom and a dad, from two parents. Well, asexual reproduction is the opposite. It's a type of reproduction where, yes, there are offspring, but they actually arise from a single parent. So there's no exchange or combining of genetic material. This creates offspring that are actually genetically identical to the parent. They contain the same number of chromosomes. They have all the same versions of the genes, everything. They're genetically identical to the original parent. Now, I was going to say something else, but I don't remember what. Oh, yeah. So think of it like a natural form of cloning. Cloning, when we hear the term cloning, we think of a lab where you take, for example, Dolly the sheep. They took a sheep and they cloned it, making a baby sheep that was genetically identical to the original sheep. Well, this is more like cloning in nature. Nature has its own way to make genetically identical offspring to the parents. There are three main types. The first is called binary fission. Now, it's actually what binary fission is. It's mitosis for single-celled organisms. Remember we said in mitosis, you double the DNA, and then you take one cell and split it into two. Well, binary fission is exactly that. It's when a single-celled organism reproduces by cell division. So you take one bacteria, it divides, you now have two bacteria. They're genetically identical to each other. Like we have here, got this little bacteria. He gathers up material, replicates his DNA, goes through mitosis, and you end up with two bacteria cells. So it's mitosis. But when you divide a cell in mitosis and you are a single-celled organism, that means you are going from one to two organisms. The next one is called budding. So this is when the daughter cell will actually grow off of the parent. Then once it gets to a certain size, it will break off and become its own organism. We see this with yeast. These are yeast cells. So here we have the yeast, and here, this little thing right there, that's a whole other yeast that's growing out of it. So imagine if you had a baby growing out of your shoulder, and then the baby broke off, and it grew up to be identical to you. We also do see this with some organisms, like different kind of simple marine invertebrates like sponges and cnidarians. Those are coral and things of that nature. So here we have a piece of coral. It's creating this little bud. The bud develops and grows, becomes a smaller version of the organism, breaks off, grows, and now you got two genetically identical organisms. And the last is called fragmentation or regeneration. And this is when an organism can regenerate or regrow after being cut from an existing parent organism. So here's a planaria. It's a small type of flatworm. But the interesting thing about these is we take this one planaria, it's a full-grown planaria, we cut it up. We have 1, 2, 3, 4, 5, 6, 7. I've cut up to 7 pieces. Well, each piece will actually grow and become its own planaria. So now I went from having one full-grown planaria to 7 itty-bitty planaria that will, take in nutrients, they will grow, and they will all grow to be full-grown planaria. And they will all be genetically identical to each other. And that's what we see here. There's just a drawing version of it. Cut it in 2, you end up with 2. You can also see this with, again, things like sponges and cnidarians, coral. You can also see this starfish and even earthworms. If you cut them at a particular point, at a particular spot, you can regrow it and have 2 starfish or 2 earthworms. However, they are a little bit more developed in terms of internal body systems. So if you just cut like the tip of the arm off, the tip is not going to regrow into a new starfish. You have to cut it off closer to the central disc. That's where a lot of the other organs are located. All right, so there's that. And that takes us to our additional resources. Woo! So first off, we have Science in Pajamas. We give you a brief explanation of the different kinds of mutations, provide some examples, and then also a video on asexual reproduction and the different versions of it, and my cute little drawings. Then in terms of other videos, we have the Amoeba Sisters talking about mutations. We have Fuse School talking about mutations. We have Don't Memorize, which is mutations and their types. We have Bozeman's science, what is cancer? We have the Amoeba sisters, asexual versus sexual reproduction. We have Fuse School, what is asexual reproduction? And we have Adventures in iSTEM, asexual reproduction, types, advantages, and disadvantages. All right, so I hope this helps to clear things up. I will post the link for this Prezi in the description, the video description, so you can have access to the additional resources. But in the meantime, you guys, just stay awesome, stay curious, and most importantly, stay safe and healthy. All right, guys, take care of yourselves.